Bismillahirrahmanirrahim. We start another chapter on the new component and some new questions on human nutrition as asked by some of you. So this is the 5090 O-level biology syllabus and this is the new syllabus for 2023. Now starting with question number 1, the liver is an organ with a large number of different functions. Figure 4.1 shows the liver's blood supply and some other organs. The blood vessels are labeled T to R. Now, the first thing to remember is that this is the aorta, right? And any vessel arising from the aorta has to be an artery. As this is going to the liver, so this will be the hepatic artery. This is just for you to understand, and I'm explaining this to you. So, aorta, A artery. And then anything which is entering the vena cava, this is the vena cava which they have labeled here. And you can see this is going to the heart. This is coming from the heart, so this has to be the aorta. This is going to the heart, so this has to be the vena cava. And anything which enters it, like this vessel here, this one here, entering it, has to be the hepatic vein. All the veins drain into the vena cava. So A aorta, A artery, V vena cava, V vein. So if it's from the kidney, it will be the renal vein. If it's from the aorta to the kidney, that will be the renal artery. So hepatic artery, hepatic vein. Renal artery, renal vein. Renal artery will arise from the aorta. Renal vein will take the blood to the inferior vena cava or the vena cava. You can also say that as well. So now what we had to say is shows the liver and its blood supply, and you can say there's gallbladder, and then there is a vessel R here. Now this vessel R is connecting the duodenum, which is the small intestine, and this is going here and it's connecting it to the liver. Now you know that which vessel connects that, that is the hepatic portal vein. Hepatic portal vein, which connects the small intestine to the liver, which is carrying all the absorbed nutrients which you have had in your meal. If you've had chicken kadai, then it's going to have amino acids. If you've had a naan, then it's going to contain glucose. So the hepatic portal vein will contain glucose, will contain amino acids, will contain water, will contain vitamins, vitamin C, vitamin D, and it will contain any calcium or iron which you have eaten. So it will be containing mineral salts. But the fats will enter through the lacteals and then will enter the lymphatics. So the hepatic portal vein will not contain any fat. This is something which you must know. Hepatic portal vein, no fats. That is going to enter through the lacteals, but will then again finally will enter the liver. Now let's follow this question. A person eats a meal containing protein and carbohydrate. So maybe we've had a chicken sandwich. Protein and a carbohydrate. Complete table 4.1 to show the blood vessel that has the highest concentration of glucose, oxygen, and urea as this meal is absorbed. Use the letter P, Q, R to identify each blood vessel. Highest concentration of glucose will be in what? If you've had a meal containing carbohydrates, the highest concentration would be in R. Why R? What we have to understand is that in R is the hepatic portal vein. This is R here. R is the hepatic portal vein which is connecting the small intestine to the liver. So as you've eaten maybe a sandwich, or you've had roti, or you've had rice, or you've had potatoes, which is carbohydrates, all that is absorbed, all that is first digested, and then it is absorbed as glucose into the blood and is going to be carried to the liver into the hepatic portal vein. So R is the hepatic portal vein. So that is going to be rich in glucose. Now oxygen, we only want to talk about P, Q, and R. So where is oxygen going to be in the highest amount? It's going to be in Q. Why Q? What is Q? Q is the hepatic artery. The hepatic artery arises from the aorta. The aorta carries oxygenated blood to the entire body. So any artery arising from the aorta is going to be rich in oxygen because the pulmonary vein has brought it back to the left atrium, and then the aorta arises from the left ventricle. So the aorta has the highest amount of oxygen in it. On all the arteries arising from the aorta, hepatic artery, renal artery, except for the pulmonary artery, which arises from the right ventricle, gastric artery, in the legs, femoral artery. So all the arteries arising from the aorta will have high oxygen content. And then the third thing they asked is urea. So where is urea made? Now then you've got to go back onto deamination. Where is urea made? Urea is made in the liver. Deamination takes place in the liver. So which is the vessel which is coming out of the liver? That is P. So as the hepatic vein comes out of the liver, all the urea which is made here in the liver is going to enter the hepatic vein. So that is why the answer to this would be P, because it's just like a river flowing outside your house. You throw something in the river is going to carry it away. So similarly, the vein, the hepatic vein, is the liver is where the urea is made. So it's going to thrown into the hepatic vein. So the hepatic vein is going to carry it away. And then it said, write the letters for the blood vessels with the highest concentration of each substance in the spaces on table 4.1, which is this table here, which we've just completed. And coming to the B part of the question, amino acids are absorbed from the small intestine and transported to the liver. Describe how the liver is involved in the metabolism of amino acids. Well, what happens to the amino acids? You ate something made of meat, fish, egg, you had an omelette, you had some kebabs, you had dal. This is all protein. It's all digested to amino acids. The amino acids enter the blood. They're taken to the liver. What happens to them there? The amino acids enter the blood and they're used to make proteins by all the body cells. Enzymes in the body cells, hemoglobin, myoglobin, antibodies, they're all protein in nature. But then the excess amino acids cannot be stored. So what is going to happen to them? Deamination. Removal of the nitrogen containing group, formation of urea, and rest of the molecule is a carbohydrate stored as glycogen or converted to fat. So all this is about deamination. Now I would like to explain deamination to you. This is an amino acid. There is an amine group or an amino group. This is the amine group. Now this is converted to ammonia, NH2, NH3, and then the ammonia is converted to urea. Now ammonia is toxic to the cells. But then there is the remaining part, which is the carbon skeleton, which is left, which has got two carbons. This is converted, this is then added to carbon dioxide and water is added, and that is converted to glucose or to acetyl CoA or ketone bodies. Well, forget this because you won't understand this in O levels. So it's converted to glucose. And then this glucose is either respired or it is converted to glycogen and it is stored in the body. So either this glucose will be respired or it will be converted to glycogen and stored in the body. So this is called deamination. Why? Because excess amino acids cannot be stored. If you eat a lot of sweet things, that glucose is converted to glycogen and stored in the liver. And if you eat too many sweet things, then it's going to be converted to fat. But the trouble is that if you eat too much of proteins, it cannot be stored. Amino acids cannot be stored. So the excess amino acid which cannot be stored have to be deaminated in the liver. So the process of deamination takes place in the liver, and this is what happens. The amino part is removed. This amino part is removed and is converted to ammonia, and then it's converted to urea, and then the urea enters the blood in the liver, and then it's taken to the kidneys. And the kidneys remove the urea from the blood. So the kidneys remove the urea from the blood. You write it from the body. That's wrong. You can't be doing that. Another diagram showing you aorta, hepatic artery, gallbladder, inferior vena cava, and the portal vein. Just a diagram to make you understand how we could give you different diagrams to be really understanding all this. So how would we word this? We'd say amino acids used to make proteins. So the liver, in the liver, the amino acids will enter the blood and will carry it all around the body. And the different cells will make proteins. They'll need enzymes. They'll make hemoglobin, myoglobin. Lymphocytes will make antibodies. Deamination will take place. Removal of the amino group will take place. Formation of urea will take place, which I've just explained to you. And the remaining portion, which is the carbohydrate, will either be respired or stored as glycogen, or even can be converted to fat, basically used for the release of energy. Then coming to the C part of the question, describe the effect on the liver of the following. Insulin released from the pancreas. How does the insulin affect the liver? It stimulates the liver cells. What is it going to do? It's going to stimulate the liver cells to absorb more glucose. And then that glucose is converted to glycogen and then stored in the liver cells. So, what is the insulin going to do? It's released from the pancreas, but finally it is going to be causing the absorption of more glucose. So, for the blood, which has suddenly been now loaded with glucose because you had a piece of chocolate cake, so too much glucose has entered the blood. That makes the blood very concentrated. Now, that glucose must be removed. So somehow this insulin is going to cause that the glucose is going to be pulled into the liver cells. It's going to absorb more of the glucose from the blood. And then that glucose cannot be stored as glucose because glucose is soluble. So it has to be converted to an insoluble molecule, which is glycogen. And then it is going to be stored as glycogen. Just like in plants, glucose is stored as starch. Here in the human cells, it is stored as glycogen in the liver cells and in the muscle cells as glycogen. So the liver cells will suddenly they will develop these new channel proteins which will result in the absorption of more glucose from the blood. And then this is going to be the enzymes present in the liver cells which will convert this glucose into glycogen for storage in the liver. So there are three mark scheme points, 1, 2, and 3, but any two of these that you would got here, 2 out of 2. Then coming to part 2 of the question, adrenaline released from the adrenal glands. How does that What does it cause or what effect does it have on the liver cell? It stimulates the liver cells to break down glycogen. Why? Because adrenaline is a fight, fright, and flight hormone. It's an anxiety hormone. You need to fight. You have a lion chasing you. So you need to fight that lion or you need to run very fast. So you need more glucose in the blood. So the glycogen reserves will be broken down and the glucose will enter the bloodstream. And then the blood will carry it around to the muscles and the muscles will respire it to release energy. Breakdown of glycogen to glucose, release of glucose or the glucose entering the bloodstream, and then of course it will be used by the body cells or the muscle cells to be aerobically respired and energy to be released. Coming to the part 3, excessive long-term consumption of alcohol. How does this affect the liver? Because of the fatty liver, then there's going to be hepatitis, there's going to be cirrhosis, liver cancer, and liver failure. So any of these, you could have given me any two of these: fatty liver, hepatitis, cirrhosis, liver cancer, liver failure. Part D, cholesterol can accumulate in the gallbladder to form gallstones. These gallstones may stop bile flowing from the liver through bile duct and into the duodenum. Explain the possible effect of gallstones on the digestion of fats. What does bile do? Bile contains bile salts. And what do the bile salts do? They emulsify fats, emulsification of fats. And they break the large globules of fat into smaller globules. This is a form of physical or mechanical digestion. And this, of course, increases the surface area for the digestion by lipase. So now that there will be no bile, because it says following the bile duct and into the duodenum, So it may stop the bile flowing. So if the bile doesn't come, it's going to digestion of fat is going to become difficult, or it's going to take longer, because now there is no bile which is going to emulsify fat. So that is why people who have gallbladder stones, I know I have them as well, I cannot eat a lot of fats, and I find it very difficult because then I feel very uncomfortable after eating something like fried pakoras or fried samosas or something like that. You see, bile contains bile salts. Bile salts emulsify fats, break large globules of fat into smaller globules. So it is sort of an explanation of emulsification. If you wrote that and you even gave me this explanation, I'd give you another mark for that. And this is a form of physical or mechanical digestion. This, of course, is going to increase the surface area for the digestion of lipase. Now, you've got to imagine this, that this is one large globule of fat. Now, the bile salts, I'm going to make them into smaller globules. Now, when they're going to make them into many smaller globules, one large globule, six smaller globules, now what is going to happen? The enzyme lipase is going to work on them. This is the enzyme lipase. So the enzyme lipase will now really be able to digest it more quickly. Lipase is chemical digestion, but emulsification of fats is physical digestion. You see, this is just breaking up the large globule into smaller globules. But then the lipase, it increases the surface area. The lipase would also work on this. I'm not saying it won't work on this. It would also work on this. But of course, then it would work, it would have to work from the outside and slowly make it smaller and smaller and smaller. But now it has got all these small, tiny pieces, so that more surface area. So the enzyme lipase can now work on it more efficiently, and it is going to be much more faster, or you can say much more quickly achieved. So the digestion of fats is going to be very fast. The bile will emulsify fats, and then of course the lipase will break down the smaller fat globules, and to finally the fat will be broken down into fatty acids and glycerol. Question number 2. Blood flows through the hepatic portal vein from some organs to the liver. Figure 2.1 shows the hepatic portal vein and these organs. Hepatic portal vein, you can see this, and I'll just color this so that everybody can see this here. This is the hepatic portal vein. It's coming from here, and they're all vessels coming from all the sides, and then it's here, and it's coming from all this side, and then you can see there's also coming from here and here, and coming from here, and then it's coming and entering into the, and it's also coming from here. Now they've sort of, this is the spleen which they have labeled it, and it's also coming from that, and they haven't labeled anything else. Now it says blood in the hepatic portal vein is deoxygenated. Explain why the blood in the hepatic portal vein is deoxygenated rather than oxygenated. Basically, it's given up its oxygen to all these organs. So all these organs have taken up the oxygen and respired, because all the capillaries join to form a vein, and the capillaries have come through all the organs. So lost the oxygen to the cells of the different organs of the body, which through which it is from which it is all the capillary networks is joining to form a vein. You see, first of all, what you have to understand is there's an artery, and the artery divides into arterioles, and then there are capillaries, and then there are veins, venules, and then vein. So basically, every organ has an artery, arteriole, capillaries, venule, vein. So why blood in the hepatic portal? Why does veins always carry deoxygenated blood? Because the capillaries have joined to form veins, and the capillaries have actually where the exchange of gases has taken place. So the oxygen is given up, and the carbon dioxide is enters the veins, and so all the oxygen is being used by the respiring tissues, has been through capillaries, through an organ, and has lost the oxygen to respiring tissues. Name 4 organs other than the spleen that are shown in figure 2.1 and from which blood flows into the hepatic portal vein. Now you can see, you have to look at the diagram and see which other organs. The hepatic portal vein is all the things which are coming. Esophagus, stomach. This is the stomach. This is the esophagus. Stomach, gallbladder. This is the gallbladder. Duodenum. This is the duodenum. Coming from there. Ileum. This is the ileum. Pancreas. This is the pancreas. And the colon or the large intestine and the rectum. So this is the colon and the large intestine and the rectum. So all these vessels, name 4 organs other than the spleen that are shown in 2.1 from which blood flows into the hepatic portal vein. You could have said any of them, any 4 of these. You could have said esophagus, stomach, gallbladder, duodenum, ileum, pancreas, colon. C. Describe the role of the hepatic portal vein in the transport of absorbed nutrients. Basically, hepatic portal vein is going to transport only glucose and amino acids and vitamins and mineral salts in solution in the plasma, transported from the small intestine, which is the site of absorption, and it is going to be taken to the liver. So it says the role of the hepatic portal vein in the transport of the absorbed substances. Okay, where is it? What is absorbed? And where is it taken to? You're going from Lahore to Multan by car or by Daewoo. Role of the hepatic portal vein in the transport of absorbed nutrients. So hepatic portal vein basically contains 5 ingredients: glucose, amino acids, vitamins and minerals in the liquid watery medium, in solution, in the plasma. Because you see the blood component, there are 2 things. Number 1 is the plasma, the liquid part, and then there are the cells in it. So in solution in the plasma from the small intestine to the liver. So there were about 5 mark scheme points and you had to give me 3. So if you wrote more, you're sure you get your 3 out of 3. Then coming to the D part of the question, explain how the liver is involved in regulating the composition of the blood and in protecting against the body against toxic substances. How the liver is involved in regulating the composition of the blood. So when you eat too many sweet things, a high glucose concentration, so the glucose is converted to glycogen in the liver, and then the low glucose concentration, when there's low glucose, like just before, now these days in Ramzan, just before Iftari, your glucose levels fall. So the glycogen is converted to glucose, and then we have the insulin and the glucagon working to help in this process. And then when the amino acids enter the liver, then they are going to be made into plasma proteins. The excess amino acids are going to be deaminated. And then what about the toxic substances? Alcohol is going to be broken down. And then you could have talked of any other toxin also being broken down, which could be dangerous for the body. So just now giving you the wordings for it. When high glucose in the blood, then glucose converted to glycogen. If low glucose, then glycogen converted to glucose by the release of insulin or glucagon by the pancreas. Amino acids are used to make plasma proteins in the liver, and excess amino acids are deaminated in the liver, or if there is the toxic substance alcohol, that is going to be broken down or is fired. Question 3. A. Describe how food is moved along the small intestine by peristalsis. Circular muscles contract behind the bolus. Muscle contraction occurs behind, so that food pushes it forward. Then the circular and the longitudinal muscles work antagonistically. Then the small intestine is lined by many villi. Figure 5.1 shows a longitudinal section of a villus. Figure 5.2 shows a cross section of the same villus at VW. The diagram is not drawn to the same scale. So everybody have a look at it. PQ. Then V and W, cross section of the same villus at VW. So we've got it here, and this is what we are seeing here. So the central would be the lacteal. This would be the lacteal here, which is this part. So which is this part here. And then of course this is the blood vessel, which we are seeing here, and here, and here, and here, and here, and here. Then this has been magnified, and you can see the cell which has got the microvilli. And this is of course the artery entering, and then the vein exiting it. Now it says name structures P, Q, and R. What is P? P is the epithelial cell. Epithelial or epithelium. You could have even said epithelium. And what is Q and R? Q is a blood capillary and R is the lacteal or epithelial cell, and Q is the blood capillary and R is the lacteal, or you could have said the lymphatic vessel. Then it says the blood that flows from S enters a vein. Name the vein that transports blood from the small intestine. Very easy. We've done it a number of times in the previous question as well. Hepatic portal vein. Hepatic portal vein. In part 3 says cell T is an example of cells that form the surface of the villi. Explain why there are many microvilli on cell T. Now microvilli means just the cell membrane is just folded. It's just one cell. So the cell membrane is folded, gives a large surface area to increase or maximize absorption by diffusion or by active transport. Because if it is by active transport, then there would be lots and lots of channel proteins here, which will be in the cell membrane. Or for diffusion also, then it's going to just pass through the cell membrane, but because it's folded, it's going to increase the surface area. So gives a large surface area of the membrane to maximize, or you can say increase absorption by diffusion or by active transport. The last part of it, some of the cells on the surface of the villi secrete mucus for protection. Suggest what the villi will need to be protected against. Enzymes. You see, the enzymes will digest its own structure. The enzymes present will digest the cells of the wall of the intestines. So the enzymes like protease and lipases, they will digest their own cells and also protect it from against the acid which is coming in from the stomach. A lot of acid is coming from the stomach into the small intestine. And then there could be physical damage of food that you have eaten, something harsh maybe in the food, some seeds or something in it which are sort of will maybe damage the lining. So physical damage. And of course, then of course we have a lot of parasites in it or named pathogens, like for instance bacteria. Cholera is caused by a bacteria invading the small intestines. So diseases of the small intestine could be bacteria, could be viruses, could be some worms. So finishing this chapter, enzymes or protease, the mucus will protect against the enzymes digesting its own cells and the stomach acid, physical damage, and parasites like worms or pathogens or toxins.